Hi everyone, I am Dr. Ayesha Farooq, and today, we are going to break down the complexity of cellular respiration into simple, actionable knowledge. Welcome to the ultimate guide on glycolysis, the fundamental pathway where chemistry meets vitality. What is glycolysis? Basically, it comprises of splitting of glucose, 6-carbon, into pyruvate, 3-carbon. It usually occurs in the cytoplasm. It ends with generation of 2 ATP and 2 NADH molecules. It is an anaerobic process that is independent of oxygen. It is a 10-step enzyme-catalyzed process. In the first step, with the help of hexokinase enzyme, glucose is converted to glucose 6-phosphate. 1 ATP is consumed during this step, as you can see in the given picture. In the second step, with the help of phosphoglucose isomerase, glucose 6-phosphate is converted to fructose 6-phosphate. In the third step, phosphofructokinase 1 consumes 1 ATP and convert fructose 6-phosphate into fructose 1,6-bisphosphate. At fourth place, aldolase enzyme works to convert fructose 1,6-bisphosphate into DHAP and glyceraldehyde 3-phosphate, G3P. Now DHAP, dihydroxyacetone phosphate, is broken down to glyceraldehyde 3-phosphate, G3P. The first 5 steps of glycolysis are regarded as the preparatory, or investment, phase, since they consume energy to convert the glucose into 2 3-carbon sugar phosphates, so here the preparatory stage ended. Step 6 of glycolysis is the oxidation and phosphorylation of glyceraldehyde 3-phosphate, G3P, into 1,3-bisphosphoglycerate, 1,3-BPG. Catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase, GAPDH. This crucial energy-yielding step reduces to and attaches an inorganic phosphate group to the sugar molecule. It is a critical ATP-generating step, substrate-level phosphorylation, in the payoff phase. During this step, the enzyme phosphoglycerate kinase transfers a high-energy phosphate group from 1,3-bisphosphoglycerate, 1,3-BPG, to a molecule of ADP, forming ATP and 3-phosphoglycerate, 3-PGA. Step 8 of glycolysis is the conversion of 3-phosphoglycerate, 3-PG, into 2-phosphoglycerate, 2-PG. This reaction is a critical isomerization step in the payoff phase of glycolysis, preparing the molecule for the next step, which dehydration. Step 9 of glycolysis is the dehydration of 2-phosphoglycerate into phosphoenol pyruvate, catalyzed by the enzyme enolase. This reversible reaction removes a water molecule, increasing the potential energy of the phosphate bond in preparation for the final ATP-generating step. This step occurs twice per glucose molecule. The 10th and final step of glycolysis is the irreversible conversion of phosphoenol pyruvate, PEP, into pyruvate. This step is catalyzed by the enzyme pyruvate kinase and produces the second ATP molecule per 3-carbon molecule through substrate-level phosphorylation. After 10 complex steps and various enzymatic reactions, what is the final result? While we produced 4 ATP molecules, remember we invested 2 at the start. This leaves us with a net gain of 2 ATP molecules for immediate energy. But that's not all. We also generated 2 NADH molecules, which carry high-energy electrons to the electron transport chain, and 2 pyruvate molecules, which are the tickets to the next big stage, the Krebs cycle. Thanks for watching. If you want to learn more, like, share and subscribe my channel, place a comment. Have a wonderful day.